Okay, so we start with a piece of lasagna. No, wait, it's a piece of cell membrane. The cell membrane is mostly composed of a phospholipid layer, but how does it work? The layer is composed of lipids like this, with polar heads and non-polar tails. But still, how does it work? Well, the lasagna is suspended in water, a polar solution. There's the inner layer with the non-polar tails that stick together, and then the polar water is happy with the polar heads. But a cell membrane isn't just one piece of lasagna, it's a ton of lasagna, with a bunch of weird stuff all over it. The stuff is important because you can use it and get in the cell, and for now let's just ignore it and say you're this pink guy, oxygen. How do you get in there? You just go right in. Yep, since you're a tiny gas and you can easily get through with passive diffusion. But what if you aren't tiny and you aren't a gas? Like this guy, glucose.
How do I get through? The carrier protein.
That's one of those weird things in the lasagna wall. So the glucose is facilitated into the cell by the carrier protein. This is passive diffusion. Now the inner cell glucose can finally be at peace. And now that other lasagna stuff. This thingy. This is the cell marker. It has little stuff that marks the cell and also tells stuff, hey, I'm a thing. So anyways, how do you get in the cell if you're one of these tough dudes, sodium and potassium? Well, you use the sodium-potassium pump, of course. This is the pump from inside the cell. It's powered by ATP, adenosine triphosphate.
Once sodium binds to the pump, it stimulates the protein pump's phosphorylation, and that means it's time to get expelled. Once that's all done and good, we can take a look outside the cell. Here's where the potassium will bond to the protein. The final result of this is a phosphate group being released and the potassium being released inside the cell. But what if you have to carry a message like this guy? This message could be instructions or just a hormone. You gotta use the receptor protein. And then the first message is transported into the cell. Now that you're in there, let's just say this is a ligand and it's gotta be opened by the second messengers. Once the message is received, it tells the proteins to get to work, and they get to work then. Also, cholesterol is an important molecule that helps the cell be less permeable to random stuff.